This is Alex Hewitt and Christoph.
Christoph, hello.
We are going to talk you through a few steps about how to get started with this competition.
In this video, we're going to talk through about a problem that we care about quite a bit, which is plastics in the ocean. Now, we didn't really know how to start with this problem. How do we find the protein that we're particularly interested in? So we figured we'd start by having a quick search. And we reckon that if you guys head to Google and type in your problem of interest plus protein or gene, you can have a look what comes up. And this newspaper article actually looks pretty good to us.
You might find that sometimes it's hard to find the key protein that might be involved in the problem, and therefore you may need to trawl through a few of the search pages before you can actually pin down a protein that's important. So looking here, this hit, it looks as though a mutant was involved in the breakdown of this big name for a chemical construct called PET. So that looks pretty exciting, and it's a powerful enzyme. So now I guess the key thing is to see if we can find out what the name of the protein is. And actually down the bottom there it's highlighted,
Alex,
PETase.
I think we've found it. Do you want to go have a look where we can find some more information about PETase to see if we can help it to break down plastics in the ocean?
Sounds corny, but let's go for it.
Alright, so we're going to go back to whichever search engine you'd like, and we're going to type in the name of the protein that we're looking for. In this case, we're looking for PETase. And so having a look here, it looks like there's a really good Wikipedia article, so we'll go ahead and open that up. Wikipedia can be a great source of information. And there's also a publication from one of the journals we can see here. So we might open that up as well to have a look a little bit later. Now, Alex, if we were interested in finding out what our protein looked like and perhaps how it worked, where would you go to have a look?
Again, that sounds very cheesy, Christoph. But if you search for, say, protein database, and then let's see what the top hit there is. So clicking there, so the protein data bank would be a good resource to hit on, and again, so you can search for your protein of interest there. And now coming up, you can see a range of different proteins. So that funny name at the top, the 5XJH, that's just a database name for the actual crystal structure of the protein. And beneath that is the paper, the research paper that describes where that data was generated from. So essentially you can click anywhere on the link and that will take you further into the actual protein crystal structure there. That cartoon on the left is displaying the structure of what this protein of interest would look like. And as Christoph scrolls down, the different domains will come into view. And so the domains are the functional aspects of the protein, and they are essentially the important parts of the protein.
And Alex, I think if you have a look a little bit further up the page, you can also see a link through to the original paper that talked about it. Is that right?
Yeah. So some journals will be hard to actually get the paper from because the publisher may require you to subscribe to the journal. If that's an issue, please just email us and we will see if we can source the paper itself. The other option is to actually email the authors of the paper, and they would often be obliging in sending a copy of the paper forward.
Perfect. So then, Alex, we've found this protein of interest. Where do we go to from here?
So I guess the key question is what exactly does the protein do and where does it come from? So first of all, answering the second question, where does it come from? Many genes and proteins are conserved across different species, so you might find that your protein of interest is actually expressed in many different species, and if that's the case, just write that in the answer box that it's expressed in many species and list a few examples. Sometimes the protein may only be found in a handful of different organisms, and if that's the case, it's a matter of writing just that name there. And so here you can see the name of the bacteria in which petase was isolated from. The next question is to what does the protein actually do? And to answer this question, you'll need to dive a bit further into the actual science behind where that protein was identified.
And so, Alex, to dive a bit deeper, you remember we opened up those Wikipedia pages in that paper at the start? Having a look, it looks like the Wikipedia article is actually pretty good at telling us how this particular protein works. And there are even some really good videos of how the protein works and what it results in the breakdown of the plastic for.
I think it's important to remember not to just cut and paste information from other people's papers or Wikipedia itself. You can certainly use this as a good reference, but it's important to write it all in your own words. But do feel free to reference or cite where you got this information from.
Yeah, it'd be great to know what you guys were thinking when you wrote your entries, and it'd be really good to read the papers that you found as well. Yeah. We also opened up another paper when we were doing that first Google search that talks about the protein, this petase, in a bit more detail. And I think in reading these papers as well, you can find out more about how the protein functions. And it will also usually in papers give you a nice background as to the science behind the protein and how it's related to the problem at large.
Yeah. One of the final key questions that we would like you to try and answer is, what are the potential adverse effects from enhancing this protein? So what would happen if we made a petase that was so good that it dissolved any plastic that it came into contact with? And to answer that question, I think you just need to brainstorm and think about the adverse effects.
Perfect. I think that's all that we had to talk about today. If you have any questions, please feel free to submit to the frequently asked questions or Ask a Scientist page on the website. They'll come through to us and we might be back with a video in 2 or 3 weeks to answer some more of your questions. Good luck and be in touch.